Okay, everybody. I think we'll start. I'd like to welcome you all to this week's seminar hosted by UCD Research Partners team as part of our COVID-19 research seminar series that we've been running since the autumn time of last year. So if anybody hasn't been to any of our seminars in the past, just to go through, why this came about and what we're trying to do. So essentially, we want to have two purposes to the series, and that's to highlight some of the great COVID-19 research that takes place across the university and its 6 colleges, and to help facilitate greater interdisciplinary collaboration within UCD and also potentially with any external partners in and around COVID-19 research. So as you might be aware, this is the time and the day of the week that we seem to always have these seminars, so please look out for them in the future. We have more next week. The format of our sessions typically start with me introducing our guest speaker, and they'll talk a little bit about their work and their research, and we'll do a little short Q&A afterwards where the audience can participate. So if you have any questions, please feel free to submit any questions via the Q&A function or the chat function at the bottom of your screen. Today our guest speaker is Dr. Grace Kenny. She's a specialist trainee in infectious diseases who has this year taken a newly established role that's been established between the US Embassy and UCD. It's the US Ireland COVID-19 Response Fellow. And we'd like to thank the US Embassy for that opportunity. It's aimed at strengthening collaboration between UCD, the clinical sites, and international partners. Grace is also working at St. Vincent's Hospital, providing clinical care to acute and post-COVID patients and recruiting and coordinating clinical care or clinical studies and in the Centre of Experimental Pathogen Host Research in UCD. She's undertaken a PhD on the immune response to COVID-19, and today she's going to talk about her work on humoral immunity to COVID-19. So I'd like to hand over to Grace now to tell us a little bit about this work. And so welcome, Grace. I'll hand it over to you.
Thank you. Let me share my screen. Okay, so as Kevin mentioned there, I'm currently undertaking a PhD in the area of immune responses to COVID-19. But my primary background is as a physician. So I'm working between St. Vincent's Hospital and the lab up in Severity. So because I'm in 2 sites, I'm involved in a few projects, but primarily today I'm going to talk about some of the work we've done into antibodies or humoral immunity around COVID-19. So antibodies play a really important role in any infectious disease. They're really important for diagnosis, and serology studies form the cornerstone of diagnosis of lots of infectious diseases. They're a key arm of the immune response to both acute infection and prevention against reinfection. And they're a target for therapy. So antibody therapies have been used for other infections like RSV and Ebola. So antibodies are always going to get a lot of attention with the emergence of SARS-CoV-2. And they certainly have, both in the popular media and in academic literature. And the way I see it is there's been 2 media narratives around COVID antibodies. So there was this first early narrative that came out around COVID antibodies disappearing really quickly and fear around reinfection. And this came out from reports in the literature around mainly patients with mild and asymptomatic infection who had an initial detectable serologic response, which then disappeared, indicating potential vulnerability to reinfection. And then there's been the second narrative, which is a bit more reassuring about antibody levels being stable. And this is from studies that have shown really most people do make a good antibody response. And these are usually stable for a number of months. But what has come out quite clearly as well in the last kind of 7 months really is the possibility of reinfection has been definitely confirmed. So this study at the bottom there is from August 2020, published in Clinical Infectious Diseases, the first confirmed reinfection with 2 genetically distinct strains of SARS-CoV-2. And there's been other reports of reinfection since then, but it's something that's still, I feel, being portrayed as quite a rare event, which isn't quite matching what we're seeing on the ground. We're seeing a number of convincing reinfections in the hospital, and I know other clinical sites are seeing similar. I've seen probably 10 to 15 convincing reinfections myself. But these aren't being reported because the bar for reporting a reinfection is to have 2 genetically distinct viruses and being able to prove that by sequencing, which is a really high bar and quite difficult to do clinically. And it was hard enough to get a test in March and April when most of these first infections happened, never mind trying to chase down that swab to get it sequenced. So it's certainly an issue, and the real burden of reinfection we're not sure about. But all that is to say, essentially what we need to have is a threshold of antibodies at which we can tell people they have a durable protection against reinfection. But we're not there yet at all with SARS-CoV-2. So we've been doing some work trying to kind of contribute to this knowledge base. So the project I've been working on a lot is one entitled Detection and Quantification of Neutralizing Antibodies Against SARS-CoV-2. So this was a grant awarded to Professor Paddy Mallon, the director of Zephyr, who's my principal supervisor of my PhD, and also Dr. Virginie Gauthier, the lead virologist in Zephyr, who's one of my PhD supervisors and others. And this was a grant from the SFI COVID-19 Rapid Response Funding Call. So this project, it has three main parts. So the first part was to screen a large number of definitely COVID positive patients to see if they're seropositive, and to identify samples to pull for the second piece of work, which is to develop a more sensitive assay to more accurately quantify antibody levels and correspondingly do a neutralizing assay, which determines functional capability of antibodies. And then the final step is to purify antibodies that we know are highly potent from the earlier work. So this is the data that came out of that first phase of the study. So we looked at 327 plasma samples from 202 COVID positive individuals confirmed by PCR. And so some patients contributed more than one sample over different time points. And then we also looked at a large number of controls, including 450 that were taken pre-2020. So these graphs are from 4 different assays. So the top 2 and the one on the bottom left are IgG-based assays, and then the one on the bottom right is an IgM-based assay. So on the x-axis, you have time from symptom onset in days. The y-axis has antibody levels in terms of antibody titers, so a signal to cutoff or a cutoff index. And the little dots are individual patients' antibody titers, and the colors correspond to disease severity. So as well as screening for the second phase of this grant, this had its own outputs. So first of all, it looked at antibody dynamics in the first 3 months post-infection. And so this is a fitted line of antibody levels over time. And you can see antibody levels are low initially, begin to rise and peak around 6 weeks, and then begin to tail off, which could indicate some early waning of post-infectious immunity. But where this study has been really useful as well as in evaluating the real world performance of these tests. So they were quite specific, so a positive test is a good rule in, but they weren't so good in terms of sensitivity. The overall sensitivity in this time period was 70%, but it depends on when you do it. So if you take an antibody level after 6 weeks post-infection, the sensitivity is around 90%. But beyond this 3-month period, there's still really not any good real-world evaluation of commercial antibody tests in the literature, which is something that we're working on. So this has been really helpful for me counseling patients. So essentially, if you have a negative or a positive antibody test, it's pretty indicative that you did have COVID-19, especially in the context of a compatible clinical syndrome. But we don't yet know what this means about protection against infection. But if you have a negative result, particularly beyond this 3-month time period, which is where we're usually using them in the hospital, it's usually patients who may have symptoms consistent with long COVID, who couldn't get a swab months ago. We really don't know what a negative result means. So it's been very useful clinically. And then the second part of the study, so the main part I've been working on is the development of this quantitative assay. So I'm using an electrochemiluminescence platform with MSD, which is a similar concept to an ELISA, but it has advantages in that it's more sensitive and has a broader dynamic range to more accurately quantify antibody levels. And we also work in a multiplex format. So I can measure 4 different antibodies against 4 different antigens in the one assay. And so you have a well that has these little dots with different antigens in them, put plasma in, the antibodies attach, and then you have a detection antibody. And then in correlation with this, Dr. Virginie Gauthier and another PhD student, Sophie O'Reilly, have been doing a neutralizing assay, which is a live virus assay or an authentic virus assay, as Zephyr has biosafety level 3 capacity. So these are the best in vitro models of neutralization that you can really get. And essentially what they do is incubate virus with serial dilutions of plasma, add them to VROE6 cells, and then determine the titer at which 50 or 90% of infection is inhibited. And they measure infectivity two ways, both with viral load in the supernatant around the cells by qRT-PCR and also counting infected cells by flow cytometry. So the fundamental output of this part of the assay is a really detailed profile of antibody responses in a large number of patients. So both in terms of antigenic targets, antibody concentrations, and functional capacity to kill virus. And we know exactly who these patients are. We know how bad their disease was. We know how far they are from symptom onset and age, comorbidities, all those things. So it helps contribute to knowing who has a better functional antibody response. But there's a number of other potential uses for an assay like this. So I think some of the most prescient would be looking at vaccine utilization. So for example, there's good data coming out now that patients who've previously had COVID-19 have a response to their first vaccine in the way someone who's COVID naive would have to their second vaccine, which makes sense. So your vaccine is kind of a booster to your infection. So we could use an assay like this to guide who only needs one vaccine response, which potentially could be a large number of people. Like if you think of healthcare professionals who've all now just been vaccinated, after the first wave in Dublin, there was about a 15 to 20% seroprevalence in healthcare workers. So potentially a large number of people who didn't need a second vaccine dose. But given the variability in antibody responses, you'd want to make sure that this was confirmed with immunoassay. We can also generally use it to monitor vaccine responses by looking at antibody levels and how they correlate with neutralizing capacity after normal vaccinations. And this is going to be important because while we know we have very good vaccines, they're not perfect, and there have been some infections after vaccination. So we could help identify people who may need either a third vaccine dose or maybe a dose of vaccine of another type of vaccine once our vaccine research gets to that point. And it can also monitor efficacy of vaccines in terms of vaccine failure. So how it can do this is because of the multiplex format of my assay. So to remind people of the structure of SARS-CoV-2, so the outside of the virus has these projections called spike, which mediate entry to the cell. And this is the target of all our vaccines. So all the vaccines make antibodies to spike. And there's also a nucleocapsid, which combines with the RNA inside the cell. And this is actually the most abundant protein. So this is the target for most of the commercial antibody assays. And so if you were vaccinated and have never been infected with COVID-19 and get a commercial antibody test, you won't actually show any antibodies. But with an assay like mine, you'll be able to show that you have spike antibodies. But if you have the appearance of nucleocapsid antibodies post-vaccination, you would know that you'd had potentially failure of your vaccination and had been infected. So there's also therapeutic implications for an assay like this. So there's two schools of antibody therapy. There's convalescent plasma and then monoclonal antibodies. So convalescent plasma, which would be plasma taken from people who've recovered from COVID-19, has been a little bit disappointing in trials in COVID-19, but some of that's to do with where it's being trialed. And this is a diagram of the phases of COVID-19. So you have your exposure to an infected person and your incubation period, which is 2 to 14 days, usually about 5 days. And during that period, your viral load begins to rise rapidly, and then it peaks usually just before symptom onset. And then you have this viral phase where you have still some ongoing viral replication, but viral load starts to decrease. So this would be when you have flu-like symptoms, fevers, chills, that sort of thing. And then most people recover, and then there's a proportion of people who develop this cytokine storm phase and will end up in hospital. And really it's your immune system that's the big player here. And this is where we need things like immunomodulatory agents and steroids and tocilizumab. And this is where a lot of the convalescent plasma trials have been based. And fundamentally where all antibody therapies are antiviral therapies, they kill virus, and where you need to be using them is where there's lots of viral replication. So really in the early viral phase or even before that. And there have been some trials looking at this early phase. So high-risk people who are just in hospital and still have mild infection, and there is some use there. But if you are going to use convalescent plasma, you need to make sure that there's actually enough antibody there to kill virus and that that antibody has a good neutralizing capacity. So you would need an assay like ours to identify potential donors. And then monoclonal antibodies, they're one of the most promising therapies really for COVID-19, but again, they need to be used early. So either in the post-exposure period or the very early viral period, or even in the pre-exposure period, maybe in high-risk individuals who haven't responded well to vaccination. And our assay can help target antibodies for monoclonal antibody development. And how it can do that is because we have such a sensitive antibody assay, what we can tell is you may have two individuals with a similar neutralizing capacity, but if one of those individuals has a lower concentration of antibodies, you know that they have more potent antibodies, and you could potentially target them for things like epitope profiling. And I do think with the emergence of ongoing emergence of new variants of SARS-CoV-2 and the fact that monoclonal antibodies only target one or two epitopes, it is going to be an area that will need continued development and continued research. So all the plasma samples that have been used in all this work have come from the main clinical study that I'm feeding into in terms of recruitment. And that's the All Ireland Infectious Diseases Cohort Study. So the PI for that is Professor Paddy Mallon, but it has multiple sites throughout Ireland. It's really expanded since the start of the pandemic. And it's the basis for lots of exciting research. But to talk a little bit about some of the other projects that I've been involved in, we've recently completed some rapid antigen test validation. So a new type of rapid antigen test and validating this in patients with confirmed COVID-19. And these are tests that I feel are probably being underutilized in Ireland. So hopefully that can advance their use here. We've been building up a post-COVID cohort, which have been a group that have been quite difficult to manage clinically and certainly need a lot more work. And we've also been building up a cohort of early COVID patients to look at biologic profiling to try and identify immune signatures that could indicate potential deterioration and maybe therapeutic targets. But there's lots of work going on with other sites as well. And there's been awards to look at obesity and adipose tissue changes in COVID-19. There's work looking at the microbiome in COVID-19. There's been other work in SEFR, such as screening for antiviral compounds. And it's a really important study and feeding into a lot of good research. So I'll finish up there, but I just want to especially acknowledge the US Embassy. It's been really great to be in this new role as the US Ireland COVID-19 Fellow, and it's great to start this valuable new collaboration.
Okay, thanks a million, Grace. It's really interesting. I really appreciate you going through all of that with us. It seems like you're involved in various different pieces of work around COVID-19 research. I see what you're mentioning there at the end, and I was just wondering in relation to the rapid antigen testing there, you mentioned, you can tell us a little bit more about what those tests are and kind of what you did with that.
Yeah, so a rapid antigen test, for anybody who doesn't know how they're used, essentially they're a bedside test for COVID-19. So you take one in a similar way to you would a normal COVID swab, so back of the throat and back of the nose, and put into a little pot of fluid and have a little cassette, which is essentially like a pregnancy test where you drop the fluid onto, and it gives you a control line, which shows that the test is working. And then if you have COVID-19, a second line to show a positive result. So what we did was looked at patients with confirmed COVID, so in both the outpatient and inpatient setting, and we took two swabs at the same time, one where we did the rapid antigen test at the bedside and then compared this to PCR. So what we showed was with this antigen test was similar to what other centres have found, was that they're really good at picking up high viral loads. So for people who are familiar with CT values, anything with a CT below 27, it picked up, and then it's quite good at ruling out high viral loads, so things above 38, and then there's a little bit in the middle where it's not as good as PCR. But really where you want to be using these tests is not for diagnosis of sick patients. So PCR is always going to be the best test for diagnosing a sick patient, but these are tests that could be used for screening, and as you get the result right away, you could very quickly remove a highly infectious patient from, or highly infectious person from a workplace, somewhere like schools or healthcare facilities.
Yeah, great stuff. I see a question coming through there from Cormac. Could you discuss the role of memory B and T cells in terms of immunity to SARS-CoV-2 infection, as there seems to be the primary mediators for immunity for many viral infections? Yeah, so obviously B and T cells play a really important role in immunity to COVID-19. But at the end of the day, on a practical level, in terms of determining a patient, an individual's immunity or an individual's vaccine response, those sort of things aren't very practical to use on a large scale. Like the only T cell based assay that I know of
that's used in clinical practice would be a Quantiferon, which is used for previous exposure to TB, which is a nice test, but it's very fiddly, for want of a better word, and on the ground it can often be taken poorly and can get indeterminate results, and it's just like serologic tests are a lot easier. And so in terms of practical application, I think antibody levels or titers are always going to be important.
Okay, great stuff. Another question come through there, Grace. Have you observed any increased severity and symptoms in patients reinfected with SARS, indicating antibody-dependent enhancement?
So I have seen patients who were sicker the second time, no one who was critically unwell, but certainly people who were sicker the second time. I can't comment as to whether or not that was antibody enhancement or if there was another pathophysiology going on. It's the truth of it. I can only report what I've seen clinically, not what's going on at a pathophysiologic level. I've seen everything. I've seen patients who've had the exact same sort of symptoms the second time. I've seen patients who had an asymptomatic infection the second time, but a very high viral load on their swab. So we were quite convinced. Yeah, patients who had milder infection the second time.
Okay, very good. A few more questions coming in through here, very popular. A recent paper showed in a small cohort that COVID patients who recovered had high antibody levels to spike protein, whereas patients who died had high antibody levels to nucleocapsid protein. Any ideas why? I
think that's a really interesting finding, actually, the kind of finding that nucleocapsid antibodies are associated with a worse response. I don't know is the short answer to that, but it's certainly something that we're going to look at further in terms of the disease severity and stuff, and the antigenic targets that we found in our cohort. It's possible that nucleocapsid antibodies just aren't as functional as spike. That spike antibodies are what you need to kill virus, and nucleocapsid just isn't as good. And they just kind of contribute to this dysfunctional immune response, this cytokine storm phase.
Okay, and on a related note, another question. Could I ask you to repeat the impact of nucleocapsid versus spike protein positivity?
So that can help us determine the difference between vaccine-mediated immunity and natural immunity. So with the vaccine-mediated immunity, you won't have any nucleocapsid antibodies, but with natural immunity, you'll have antibodies to both spike and nucleocapsid if you've had an antibody response.
Yeah, that's interesting. Just to go back to one of the other studies you were talking about in the post-COVID cohort, I was just wondering, was there any sort of particular patient type you've seen associated with that cohort? And would any of these patients be the ones that are quite sick in ICU?
Yeah, so when we set up our post-COVID clinic back in May, we kind of felt it was going to be meant for a more structured follow-up of hospitalized patients. But that's not really where the demand has been. Actually, most of our patients are people who'd had mild disease initially and kind of young, previously healthy patients. The median age of our cohort is about 47, but we've seen patients as young as 16. And they're usually people, about 3/4 of our patients had a mild disease. So they're not the cohort that had some of the large cohort studies published about them, patients who were hospitalized, that you could probably understand a bit more why they were still experiencing symptoms. But these are people who may have just had something like a head cold or a bit of anosmia, and then maybe coming out of isolation or a week or two later start developing other symptoms, things like disabling fatigue, palpitations, chest pain, and they've been difficult to manage, and there's a lot of morbidity associated. Like the median length of time off work that our patients have had is 12 weeks. It's quite a long time.
Okay. Do we know the risk of reinfection with reduction of antibodies over time?
Nope, I don't think.
Higher risk with variants?
Say again?
Is there a higher risk with the variants?
It looks like there will be a higher risk with the variants. Yeah, that's certainly one of the theories behind what's happened in Manaus, where they had a really bad first wave, really high seroprevalence after the first wave, and now have had a second wave which is worse than the first wave, and you would assume that that's something to do with immune escape with the new variants.
There's a statement here that asking for your comment on is neutralizing antibodies are important. However, non-neutralizing antibodies are also an important function of humoral immunity in vivo. What's your take on that?
So what's my take on, repeat the question, repeat the statement again?
neutralizing antibodies are important. However, non-neutralizing antibodies are an important function of humoral immunity in vivo.
Yeah, so I suppose that's something that we'll be looking at a little bit more, where focus is on the neutralizing capacity of antibodies, but we'll know what sort of patients have high titers of antibodies or high concentrations of antibodies that don't necessarily have good neutralizing capacity, and the sort of phenotype that might be associated with that.
Okay. Another question on antibody levels in terms of the comparison between post-infectious patients and post-vaccination. How do you compare? Do you see any differences between antibody levels between those two?
So we haven't been looking at vaccinated plasma so much at the moment. We've been focusing on convalescent plasma, but my understanding from some other researchers is that there is less of a variation in neutralizing capacity with vaccinated plasma compared to convalescent plasma. There's a really broad range of neutralizing capacity in convalescent plasma. Some people will have virtually no neutralizing capacity, where that difference is smaller in vaccinated plasma, and generally everybody has a more detectable neutralizing capacity.
Okay. We're just coming to a close here now. I was just wondering if anybody had any other comments or questions they wanted to put to place. If not, we can always continue the questions through the COVID-19 research Basecamp team that I believe you would have signed up to when registering. So of course, feel free to use that if something comes to mind after the event. But on that note, I think I might bring it to a close. And thank you very much, Grace, for your time and joining us today and talking about your work. Sounds like an awful lot's going on. Also, thanks to all the attendees for all your interest and your questions and your time as well. And again, please go to the COVID-19 Basecamp if you'd like to continue any conversations. Next week, we'll have another seminar on at the same time on Wednesday. We'll have two speakers. We'll have Professor Susie Geiger and Dr. Laria Galasso, who will explore the role of solidarity in the management of COVID-19 in the pandemic in terms of the Irish context, which will be a qualitative study on the citizens' experiences during COVID-19. So on that note, thank you very much again, Grace, and for everybody who attended, and I hope everybody has a great day.